Have you ever wondered how a thought just happens? Or how you can instantly remember the smell of your childhood home? Well, it all comes down to this incredible, silent, and super fast language that the cells in your brain are using right now. Today, we're diving into the absolute foundation of that language, synaptic transmission.
To really get a grip on this, you have to think about the scale we're dealing with. I mean, the human brain has something like 86 billion neurons. That's, more than 10 times the number of people on the entire planet, all packed inside your head. Getting that many individuals to talk to each other is, frankly, one of the most mind-blowing feats of engineering in the universe.
So, what's the solution to this gigantic communication problem? It's a tiny, brilliant little structure called the synapse. Think of it as the point of contact, the microscopic handoff point where one neuron passes a message to the next. It's what forms all the intricate circuits that, make you.
Okay, so here's our game plan for this explainer. First, we'll think about the brain as a kind of giant social network. Then, we'll compare the two main ways neurons chat, the direct line versus the messenger. After that, we'll get into the nitty-gritty of a chemical message, see how those messages are packaged, and finally, we'll uncover the secret trigger that lets them fly. Right, so let's stick with that idea of a social network. You've got 86 billion users, your neurons. The synapses are all the connections between them. The DMs, the follows, the group chats that let information zip around. This is the network that's processing everything you see and hear, forming your memories, and behind your every single action. And here we go, this is the heart of that network. This is a really great illustration of a chemical synapse, which is by far the most common type in your brain. You can see the top part, the presynaptic terminal, just jam-packed with these little bubbles full of chemical messengers, and down at the bottom is the receiving neuron. Now they're super close, but look carefully, there's a tiny little gap between them. So it turns out, neurons actually have two main ways of talking to each other. You can kind of think of it like this. You either have a direct, hardwired phone line, or you send a message with a courier. One is built for pure, unadulterated speed, and the other is all about complexity and control. And those are our electrical and chemical synapses.
In this slide just shows you the physical difference perfectly. I mean, look at this. On the left, in the electrical synapse, the two cells are basically fused together. But on the right, the chemical synapse, you can see that clear gap between them. That little space is called the synaptic cleft.
And this chart really lays out how that one structural difference leads to huge functional differences. With the electrical synapse, that physical link means the current just flows right through. It's almost instantaneous. But the chemical synapse, it needs a messenger to actually cross that gap. That introduces a tiny delay, but that delay is everything. It's a point of control, a place to fine-tune the signal, which allows for way more complex communication.
So let's just zoom in on that direct line for a second. That physical bridge is made of these protein channels called connexons. They form a literal pore between the two cells. This allows ions to just pour through, making the signal incredibly fast. And this is absolutely essential for things that need to be perfectly synchronized, like the neurons in your brainstem that control your breathing. You really don't want a delay there. Okay, so electrical synapses are fast and cool, but the vast majority of synapses in your brain are chemical. They're a little more complicated, but that complexity is what allows for this rich, nuanced communication. So let's break down exactly how this chemical message gets sent step by step. Now this diagram shows the whole shebang. And I know it might look a little intimidating at first, but this whole elegant dance happens in a fraction of a second. We're going to break it down into 5 key steps to make it super clear.
All right, let's walk through it. Step 1. An electrical signal, the action potential, zips down the neuron and slams into the presynaptic terminal. Step 2. That arrival forces open special gates called voltage-gated calcium channels. Step 3. Calcium ions flood into the cell, and this causes these little sacs, or vesicles, that are filled with neurotransmitters to merge with the cell membrane and dump their contents out. Step 4. Those neurotransmitters float across the gap and lock onto receptors on the other side. And finally, step 5, the neurotransmitter gets cleared out really fast, which ends the signal and gets the synapse ready for the next one.
So we've just said that neurotransmitters are released from these vesicles, right? But how does that actually work? This question leads us to one of the most fundamental ideas in all of neuroscience, something called quantal release.
This was the big mystery that pioneers like Bernard Katz were trying to solve back in the 50s. When a neuron sends a message, does it just dump a random amount of chemicals out there, or is it more precise? Is it an unregulated flood, or is the message sent in nice, neat, standardized packets?
So what they discovered, by looking at the connection between nerves and muscles, was that even when nothing was happening, the muscle cell would show these tiny, random little electrical blips. But when they stimulated the nerve, the big signal they got was always a multiple of that tiny unit. It wasn't random at all. This led them to this incredible conclusion here. The big signals were just made up of a bunch of these identical, smaller ones. And here's the key takeaway. Neurotransmitter release isn't a continuous flow. It's discrete. It happens in these standardized packets that scientists call quanta. So the neuron isn't just shouting at different volumes, it's sending a specific number of identical messages.
So what exactly is a quantum? Well, it's the contents of one of these things, a single synaptic vesicle. Just look at this micrograph. Each one of those little circles is a vesicle. It's like a pre-packaged message in a bottle, stuffed with thousands of neurotransmitter molecules, all ready to be sent.
And here is the smoking gun. This is the direct visual proof. On the left, you see the vesicles all lined up, docked, and ready to go. Then on the right, after the neuron fires, you can literally see these little omega-shaped pits where the vesicles have fused with the membrane and released their contents. It's just a beautiful illustration of quanta release in action.
Okay, so we've got the what, the vesicles, and we've got the how they fuse and release these quanta, but there's still one massive piece of the puzzle missing, the why. What is the specific, final trigger that tells those vesicles to fuse at that exact moment?
Let's think back to step 2 of our process. That electrical signal, the action potential, arrives. Its main job isn't to physically push the vesicles. No, its job is to open a very specific gate, a voltage-gated channel that lets one particular ion come rushing into the cell. And that flood of ions, that's the final trigger.
And that critical ion is calcium. This image is just stunning. Scientists used a dye that glows when calcium is around. So when the neuron fires, you can actually see this gorgeous wave of high calcium concentration, that's the bright colors, flooding right into the terminal. You're literally watching the trigger being pulled.
And this, right here, is the bottom line. This is the conclusion from decades of brilliant research. A rise in calcium inside that presynaptic terminal is both necessary, meaning if you block it, nothing happens, and it's sufficient, meaning if you just inject calcium in there yourself, you can make the neuron release neurotransmitters without any electrical signal at all. Calcium is the one non-negotiable command to go.
So, we've gone on this pretty wild journey, right? From an electrical signal to a flood of calcium to these tiny vesicles releasing perfectly measured chemical packets. And this happens billions of times a second across trillions of connections in your brain, which kind of leaves us with one last huge question. If this microscopic chemical dance is the foundation for everything, every thought, every memory, every action, how on earth does it all scale up to build the entire rich, seamless world that we experience every single day?